Good evening. I am Faria. So basically, I'll be presenting the topic immunoresponses to fungi, immune responses to fungi. So let's begin. Fungi are eukaryotic microorganisms. Eukaryotes are organisms whose cells contain a nucleus and other membrane bound organelles. There are about 1.5 to 5 million species of fungi which have the ability to grow almost anywhere, including the ocean, soil, plants, and animals. Some form spores, basically which we inhale on a daily basis. For example, we can take the case of Aspergillus species, and others live as commensal organisms. For example, the Candida species. Despite the close encounters we have with fungi, however, our immune system, it recognizes only, not completely all types of fungi. So our immune system recognizes and protects us from fungal pathogens, but the concept is not well understood as compared to bacteria or viruses. Although there are a huge number of fungi in the world, only 400 types are potential agents of human disease. Most of the disease-causing fungi are opportunistic pathogens, meaning they only cause disease under certain circumstances, such as when the system becomes weakened. For example, we can take the case of patients suffering from HIV or cancer, etc. Next, moving on to the next slide. So basically, fungus have various ways of entering a human body. So it can be, the main organ through which we breathe, inhalation, is our nose. So entry by inhalation. Fungus can enter human body, and the fungal spores can basically enter our body through our nostrils. Next is, human beings are basically bound to injuries. If you have an injury, say for example, on your palm or your hand, it can easily enter your circulation through the damage which has been caused to your skin. Also, it can enter human gut, mucosa, or other mucal linings. Why? Because fungi are opportunistic, and they are opportunistic pathogens. And when given an opportunity, they do not miss. They find multiple modes of entry into the human body. Fungal diseases or mycosis are classified based on certain criteria. The first is site of infection. So the site of infection, whether it is superficial, it can be cutaneous, it can be subcutaneous, or deep and systematic. We can see the diagram here. This is the diagram of a skin. It can just go through superficial layers also, and it can enter the deep layers as well. Next is route of acquisition. It can be endogenous or exogenous. Basically, it can be through inhalation, or it can already exist in the gut microbiota or gut lining. Next is virulence. It can be primary or opportunistic. Like we discussed, fungal species are opportunistic pathogens. Next, the type of immune responses. Basically, there are immune responses or types of immunity can be of two types. The first is innate, inborn, or non-specific defense mechanism. So innate immunity is non-specific. The second one is acquired, adaptive, or specific defense mechanism. And this is the third line of defense. So innate immunity can be further divided into external defense and internal defense. So external defense is the first line of defense. Internal defense is the second line of defense. In external line of defense, it can be physical barriers like skin, mucus, nasal hair, cilia. Chemical barriers like oil glands and sweat by sebaceous glands, stomach acid, or low pH, that is the gastric juice, cerumen, earwax. Cerumen is also called as earwax, lysozyme in tears and tissue fluids, vaginal bacterial producing lactic acid, which are low in pH. Next is internal defense, which is also known as second line of defense. It may be phagocytes that include macrophages, and WBCs that include neutrophils and monocytes. Inflammatory reactions, fever, interferons, complement system, natural killer cells, also known as NK cells. Now moving on to acquired, adaptive, or specific defense mechanism. It can be of two types, active immunity and passive immunity. In active immunity, it comes in contact with antigen, that is T cells, cytotoxic T cells, plus helper T cells, plus suppressor T cells, plus memory T cells. In B cells, we have plasma cells and memory B cells. And third is antigen presenting cells. Those can be macrophages, B cells, and dendritic cells. Next, coming on to passive immunity. Antibodies artificially produced outside or they are directly injected to the body. So basically there is no contact with pathogen. Now moving on to neutrophils. We all know that neutrophils are one of the most important components of innate immunity, which play a very vital or defensive role against most fungal species. And the neutrophils are highly phagocytic. So neutrophil, here we'll discuss in this slide about neutrophil extracellular traps in fungal infection. So neutrophils play a pivotal role in the defense against fungal pathogens. These phagocytic hunters migrate towards invading fungal microorganisms and eradicate them by phagocytosis, oxidative burst, and release of neutrophil extracellular traps. And these are called as NETs. So NETs are fibrous. They have a three-dimensional network structures composed of nucleic acid and various granular proteins that neutrophils can release out of the cells in response to various stimuli. This structure traps pathogens such as bacteria, fungi, and it limits their spread through the body and kills them with high concentrations of toxic proteins. Basically, in the last decade, the process of NET formation has received unparalleled attention, with numerous studies revealing the relevance of this neutrophil function for control of various mycoses. Here in this figure, we can see the formation of NETs and the fungal strategies against NETs. So NETs can be produced through NETosis or rapid release from live cells. The NETosis process includes disassembly of nuclear envelope, chromatin decondensation, plasma membrane rupture, and NET release. Whereas rapid release from live cells requires cell degranulation and ejection of mitochondrial DNA, then the NETs will be assembled outside the cell. For fungi, they can resist NETs by cell wall components and secrete enzymes to help them escape the NETs. Now coming to cell wall components. These are the cell wall components that are medically important. There are three types. Glucans. It can be the polymers of glucose. Next, we have mannans, long chains of mannose. Chitin, a polymer of N-acetyl glucosamine. So these together form PAMP. So PAMP is pathogen-associated molecular patterns. So next, now we come to the innate recognition of fungi by the immune system, or we can say they are the key PRRs recognizing fungal PAMP. Fungi are recognized by the cells of the innate immunity system, for example, dendritic cells and macrophages, which binds together components of fungal cells, cell walls using pattern recognizing receptors, PRRs, on their surface. So C-type lectin receptors, which are called as CLRs, for example, dectin-1, are particularly PRRs in antifungal immunity, although several other PRRs are involved, including the toll-like receptors, which are also called as TLRs, example, TLR2. When PRRs bind fungi, they signal using their intracellular tails or associated molecules. FR gamma resulting in phagocytosis, initiating of killing mechanism. Excuse me, sir. I'm sorry, sir, I will continue. So when PRRs bind fungi, they signal using their intracellular tails or associated molecules, FCR gamma, resulting in phagocytosis. Initiation of killing mechanism, for example, production of a reactive oxygen species, and also help drive the development of adaptive immunity. Adaptive immunity to fungi is only partially understood, although it seems that CD4 plus T cells that make IFN gamma, that is TH1 as an example, or IL-17, TH17, provide the best protection during fungal infections, as these help drive effective killing by innate effectors such as neutrophils and macrophages. So here we can see in the diagram our dendritic cell decorated with antifungal PRRs. Upon recognition of fungi through these receptors, signaling results in generation of TH1 by 17 in adaptive immune responses and activation of innate effector cells. Now, finally, coming to our last slide. Most fungal infections are common in human beings. Pathogenic fungi are opportunistic, as we discussed in the earlier slides, but can cause fungal infection disease in patients with immunocompromised conditions, such as malignancy, chemotherapy, transplantation, acquired immunodeficiency syndrome, and usage of immunosuppressant drugs. Most invasive infections we know are caused by Aspergillus species, mucormycetes, Cryptococcus species, and Candida species. For instance, we can take the example of AIDS patients who basically suffer increased incidences of mucosal candidiasis, histoplasmosis, coccidioidomycosis, and cryptococcosis, and other fungal infections, which give an indication that patients which are more susceptible infections as the number of T helper cells are low, which means that fungal immunity against fungal infection involves T helper cell mediated responses, and the body which is compromised due to lack of B cells are not quite susceptible to fungal infections. Further, it also gives us understanding that adaptive immune components like T cells are crucial for fungal elimination and not the B cell response. Th1 subpopulation of cells secrete interferon gamma, which activates macrophages, which leads to engulfment of the fungus, thereby playing a crucial role against eliminating the fungus from our system. Thank you all.